I'm Kevin Elmy. In this presentation, Outside of the Box Farming, we're going to go through kind of my farming career in the last 22 years. I went through the University of Saskatchewan, through the College of Ag Culture. I've been trained as a traditional agronomist. I've worked in traditional jobs. When I moved back to the farm and started doing these practices and seeing where the risk was, I realized we need to do change. And this change is going to challenge a lot of modern agricultural methodology. And hopefully, when it's done, hopefully you do have some questions of reflect on how we can do changes in our own operations. To give you a little bit of background, Friendly Acres Seed Farm was started in 1939 by my grandparents. We farm 1,500 acres. We do seed production. We do seed retail. I do some consulting. I do public speaking. Plus, I'm a writer, and now I'm an author. On our farm, we have grown a very diverse number of crops. And when we start talking about just the crop types, when you look at something like corn, we've probably grown 400 different varieties on our farm just to see which ones perform well, and they were based on corn grazing, how the cows graze them. So we were doing yield trial, we were doing complete feed test on it, and then we were seeing palatability by the cows. So each one of these, some of these we've grown for a longer period of time than others, but it was something that we would try and just see what fit in a rotation and how to manage them. And this is where one of the things we really need to do is we really need to take a portion of our farm and do something different on it. And that's that 5% rule. So take 5% of your acres and do something different with it. And this may be a seeding rate trial. It could be fertilizer. It could be whatever. And in this case, in the picture, what we did is we contacted local aerial applicator, and we got him to fly on some tillage radish into our standing triticale crop. We wanted to see how it performed. And I'm a big believer in learning by doing. When you do it, you're going to learn a lot more than learning from someone else when they say it didn't work. The other thing that when we start talking about this 5% rule, we have to remember that 1 year doesn't make a trial. We need to see, okay, so if we do it and it's a really dry year of seeding these radishes in, well, of course it's not going to work because we didn't get rain. So if you tried it for 3 years and it didn't work, okay, most likely it didn't work, or there's something that wasn't done right. So, that learning by doing is big. We want to take a look at our operation, and we want to look at our weaknesses. That's the ugly part of managing is we never want to see the bad stuff. We always want to be on looking at the good stuff. But in order to, the links of a chain, the chain is only as strong as the weakest link. So take a look at that. Look for those weak links and how do we improve them. The other thing that I really stress to producers is ignore your gross dollars per acre. Gross dollars per acre means nothing. What is really important is your net dollars per acre. That is what you get to keep at the end of the day. The other thing I tell people is, okay, yeah, it's great to see that those dollars per acre in that year, but what does the net dollars per acre look at in a rotation? That is, once again, really important, because having a crop that maybe doesn't give a big net on a year, it may, whether it's fixing nitrogen, building mycorrhizae, fungi in your soil, whatever, maybe it's contributing later on. And the example I use in here, so I've done 2 different rotations. One rotation and one crop oscillation. So in a rotation, I've used an example of soybeans using a full season cover crop that we're just letting it grow and not doing anything with. We're then having the perennial ryegrass seeded into that cover crop, and then this way we get 2 crops out of 2 years of cover with one seeding. And then when we take the perennial ryegrass off, in year 4 we go to a full season cover crop that will hay. So that is a rotation. It's not very sexy. It's not glamorous. as compared to the crop oscillation over on the right hand side, where we look at the canola wheat crop oscillation that is very popular in areas. And so the neat thing with this is just budgets and using some long-term averages. And that's the other thing I stress people is don't get hung up on, okay, yeah, the canola went 65 bushels an acre this year. What is your 15-year average? It's probably not that high. So when we're going through this, so I've put in prices for seed, inoculants, fertilizer, fungicides, so your cash costs. Then in the next section we have our seeding, harvesting, spraying, land rolling, or swathing costs to figure out what our costs are, our total expenses. And when we look at this 4-year rotation, our expenses, our average per acre is $86.50, whereas the wheat canola crop oscillation, your 2-year average is $276.50. When we get into the yield, so 30 bushel soybeans, 900 pounds of perennial ryegrass, and then 3,000 pounds of a cover crop. Okay, so there's your prices, there's your income, your net. When you compare the 2 years of net, or the 4 years versus 2 years, you're looking at $119.75 versus $123.50. So when you look at your return on investment, your ROI, that $119.75 and only spending $86.50, you're making way more money on that. The other really neat thing is, okay, so that cover crop in year 2, if you decided you're going to do some custom grazing of some animals, some livestock, now we're starting to, put a $50 an acre, $60 an acre grazing income off of it. That 4-year rotation is significantly above that canola wheat. So these are the things we want to look at. It's really important on the farm survival. So one of the things that we've been involved with, and indirectly, because when I started farming, the land I purchased, it was burned and baled for 50 years, so it was just like rock. So in that case, I did not buy soil; I bought dirt, and so I started doing things differently. And so I've been doing this regenerative ag idea for 20, 21 years, 22 years. So what is regenerative ag? And regenerative ag is a system of rebuilding the soils to get closer to what these soils look like before it was brought into agriculture production. We want to restore natural ecosystems in our soils. And by doing that, we're going to restore our carbon, water, and nutrient cycles. When we start looking at these principles of regenerative ag, you'll see a whole bunch of different ones. So, I call these the Omni 5 principles. And the first one that is the easiest to get into your rotation or into your management is having a living root in the vegetative plant stage throughout that growing season. It's really important to do that. We want to increase our plant diversity that we're growing in rotation. We want to reduce our tillage. We want to reduce the use of synthetics, and we want to have livestock integration. Those are the principles. You can take a look at it. You can say, well, no, I don't want to do this. I don't want to do that. Just if you the more of these principles you use, the quicker these soils will repair themselves. Soil development in the prairies, where our soils were developed with a co-evolution of plants growing and animals grazing them. Today, our modern agriculture relies on chemistry. Our fertilizers, our herbicides, our fungicides, insecticides, all of those things. And the really scary part is that last 6 letters, the sides, that means kill. And so when we start talking about killing, is that what we want to do in agriculture, or do we want to, build life? And so that's one of the challenges. So this reliance on synthetics and tillage, the issue that the the ecosystem has is it when we start using a lot of synthetics and using lots of tillage, it reduces our fungi populations in our soils. It increases our bacterial populations in the soils. And when we do that, that drives free soil nitrate in our soil. So when we have that, that's going to promote more weed production, growth. We're going to see more lodging. We're going to see diseases. We're going to see insects. When I was going through university, one of the things that it was thought was the way you build soil organic matter was you grow this crop and you worked it back into the soil, and that's how you build organic matter. The straw is good armor, but 75% of the carbon above the ground, so in the straw and the leaves and the grain, is lost within, on average, about 4 years. So all that carbon on the in that above ground plant is lost. Soil organic matter is built through soil microbes and root exudates, which for a lot of people is a real mind bend. So one of the options that we have is including this I guess thought or management process of cover cropping. And basically cover cropping is growing plants to cover that soil to help improve that soil. What I've learned is when we are, decide we're going to get into cover cropping, goal setting is so important. We need to know what are our issues, what are we trying to fix. Basically, what do you want to see in changes in your soil? Do you see erosion, so you want to reduce erosion? Do you see sitting water, so we want to increase our water infiltration in our soil? Do we want to reduce the weeds, insects, and diseases? Do we want to cycle nutrients? Or do we want to create feed for livestock? What do we want to do? We also need to identify, is the issue a symptom or is it a cause? The difference being, if we're solving a symptom, that problem is going to reoccur. If we solve the cause of the issue, the problem goes away. So one of the books that I highly recommend for people to read, a nice simple book. It's 140 pages long, so nice easy read. When Weeds Talk by J.L. McCammon. And so in there he explains the soil conditions that these plants want to grow in. So in this case, wild oats, we go out and we spray them and spray them and spray them, and every year they keep coming back. And one of the easy ways to get rid of it is increase the fungal to bacteria ratio in the soil. We want to tie up those nitrates, increase our calcium supply, the flux rates in that soil, and those wild oats magically disappear. And I use the magically in quotes because those are the conditions that wild oat does not want to grow in, so they do not grow. So now, instead of spraying the symptom, we actually start attacking the cause of the issue, and that causes that problem to go away. So some of the fundamentals of cover cropping: plant diversity. When we look at plant diversity, we're not just talking about the different varieties. We're not talking about we're going to mix for our green feed oats and barley and wheat, and then we have diversity. Those are in the same functional plant groups. So when we start talking functional plant groups, we're going to start looking at, number one, our grasses, our legumes, and our broadleaves as general groups. We're going to have warm and cool seasons species of each of those in each. And we have annual, biennial, and perennial, again, in each one of those groups. We want to keep that living root of a vegetative plant throughout that whole growing season. We want to reduce tillage. We want to reduce the use of synthetics, and it gives us an opportunity to integrate livestock onto our land. So as a visual, done up just this neat little triangles. So there's our grasses on the top, our legumes are on the right, and on the left there's our broadleaf. And because the broadleaves are so diverse in functional plant groups, I broke it down into brassicas, non-brassicas, and forbs. So the more of these we can include in a cover crop mix, the different root exudates we're going to be releasing into the soil. We're going to wake up more soil biology. We're going to have healthier functioning soils. So as an example, using what I call a relay cover crop. So a relay cover crop is going to be seeded with a cash crop. And in this example, we're going to use wheat. So we're going to add Italian ryegrass and subterranean clover in with the wheat. And whether we put it right with the wheat at the same time or delay it a little bit, doesn't matter. But simplicity wise, it can be seeded all at the same time. So your wheat is a cool season annual. Our Italian ryegrass is a cool season biennial, and our subterranean clover is a low-growing annual clover, cool season clover. So in this case, we have three different functional groups. We have a cool season annual, cool season biennial, and a cool season legume. So then you're able to go in, harvest your wheat, and then the Italian ryegrass and the subterranean clover continues to grow right up until freeze up and they winter kill. So this way, underneath that wheat, you can see in the picture, you can see that little bit of green underneath the canopy. Really important for building soil health. So the whole thought process behind it is the Italian ryegrass is going to tie up nitrates. When you have a grass that's only growing, 10 centimeters tall, how much nitrogen does that plant need, or water? Very little. So what'll happen is when you have nitrate in the soil and you don't have something to take it up, then the weeds start kicking in. In this case, the Italian ryegrass, it's going to soak up that nitrate because nitrate's passively taken up through water uptake. That Italian ryegrass then converts that nitrate into ammonium, amino acids, and protein. Because it's only a small plant, the protein only wants to get so high in it. So what the Italian ryegrass will do is it'll release that back into the soil as amino acids, ammonium, or proteins, and your cash crop is going to be able to take it up. One of the things when you're looking at nitrate uptake by a plant, it has to go through two different conversions. It has to get reduced to get down into a spot where it can be converted into amino acids and protein. When it's in the ammonium or amino acid or protein stage and that wheat crop takes it up, it goes straight to protein. It doesn't have to get converted. So this way the wheat doesn't have to spend a lot of energy converting it. It's already in a form it can use. So when we have this, we tie up that nitrate and we have ammonium and amino acids and protein in the soil, our weeds cannot handle that. So this way we'll see less annual weeds and we'll see less winter annuals because we have that green plant growing right up until fall. If you spray it out, now you're going to have those plants rot. As they rot, you don't have anything there to use that ammonium, amino acid, and protein. So then it breaks down back to nitrate. Now we get our flush of winter annual weeds. The subterranean clover, on the other hand, it's a low-growing clover. So this is a picture of it on the screen. So it's low-growing, kind of covers the ground, fixes nitrogen, builds mycorrhizae, and it only grows a couple centimeters high, so it will not interfere with harvest. And so whether it's wheat, barley, oats, flax, there's a lot of different opportunities we could be doing with these relay cover crops. And then if we decide that we want to do some grazing, then we'd increase the rate of the Italian ryegrass to justify, to have enough biomass there to justify grazing. So if we're in a feed situation, so we're going to use a hay and slash grazing blend. Using an oat pea mix, that's good. It's better than just straight oats, say. So with the oats, you have a cool season annual grass, and peas, you have a cool season annual legume. Now, if we add some Italian ryegrass, which is a cool season biennial grass, okay, so that's a win. If we add some Japanese millet, which is a warm season annual grass, that's another functional group. We add some Persian clover, which is a cool season annual legume, so same as pea, but little different. And then we can add some Phacelia, which is a cool season annual, in this case, forb or broadleaf. If we want something to overwinter, we can put in some chicory, which is a cool season biennial broadleaf. We can add some sweet clover, which is a cool season biennial legume, and some sunflowers, which is a warm season annual broadleaf. So once again, we're checking off this diversity in these mixes. And when we start feeding this, the animals are going to do so much better because they're going to have different types of nutrition in this ration that we're going to be bailing up. Which then brings us to the soil food web, and basically this is how you know these nutrients get cycled through. So there's lots of arrows in here moving back and forth, but basically it's starts off with your bacteria and your fungi, and then you have your predators above there, and as we keep going, we get this nutrient cycling. If we didn't have the fungi and the bacteria, this is when we have that thatch buildup, and it doesn't rot. When it doesn't rot, we don't have nutrient cycling. We get nutrient tie-up. Really important. And once again, if we just had the fungi and the bacteria, they break the stuff down, but we need something to eat them to then poop them out to get these nutrients back into the form where the plant can use. 90% of the nutrients that ended up into the plant have to go through the soil biology. So the soil biology, when we're adding fertilizer, it still has to go through these microbes. So your fungi and your bacteria, they're basically your gatherers. They'll go down and they'll break down your soil organic matter, your manure, your plant material, dead gophers, whatever else is in there. They'll start breaking that down. Your protozoa, amoeba, nematodes, those are your hunters. So they'll hunt and kill and eat your fungi and the bacteria. The other thing is when you start looking at where these fungi and bacteria are situated, they're usually within 2 millimeters of that root. So all of this activity is happening ideally right around that root. So the more roots you have, the longer you have an active root with root exudates, the more activity you're going to have around them. And you're going to get more things happening, more good things happening. One of the things that I've learned is that's really important is learning the fungal to bacterial ratio in your soils. So it's a lab test, and once you have some experience, then you get to identify, where you can eyeball fields. You can get a real good idea of where things are at without getting a test. But it's a good place to start. So what it does is it does an estimate of the weight ratio of the fungal population to your bacterial population in your soil. So with that ratio, it signals what plants want to grow and whether you're going to have nitrate dominated soil or ammonium dominated soil. This all drives plant succession. What do I mean by that? So when we have a nice little chart like this, so we can follow through. So when we have in this case when we have just bare parent material, that's all bacterial. Then you start getting some fungi in there. When most of our farmland, we're farming it in the fungal bacterial ratio of that 0.1 to 0.2. This is where we see our weeds. This is where we see high nitrates, and this is where we start seeing lack of oxygen. Now, lack of oxygen, a good symptom of that is dandelions and thistles. Once we get into 0.3 fungal to bacteria ratio, this is when farming becomes fun. Now we start to build more ammonium in our soil because we have a stronger component of fungi in our soil, and the fungi have some anti-reducing nitrate properties. So this way, it keeps it in that ammonium stage longer. Then, as as we keep building fungi, we can get more and more crops. So if you're going to be growing Sicer milk veg, and you complain that it's really slow establishing, and you've seeded it on canola stubble where you're at a 0.2 fungal bacteria ratio, Sicer milk veg likes to be about 0.5. So it's really slow establishing in the crop, but once it starts getting established, it takes off. Now, when we start going through in a 0.75, then we see our mid grasses, clovers, vegetables, 1-to-1, late successional grasses, row crops, and native plants. And then when we go over that 1-to-1, so we get that 2-to-1, this is when we start seeing things like shrubs, rose bushes, things like that start showing up. So if you have a pasture that is losing productivity, You start seeing species like that, that means that your fungal to bacteria ratio is past that 1 to 1. Once we start getting to 5 to 1, this is when we start seeing poplar trees show up, and when we see spruce trees, pine trees starting to show up. Now we're in that 100 to 1. So as we manage our soils, our land, it really influences where that fungal to bacteria ratio is. So as we have a field and we seed it down to a hay field, so we're in a fungal to bacteria ratio of zero point 0.2. We seed down a forage stand and we use a cover crop of oats. And that first year we have that oats because it's a nurse crop, and we're going to hay it off, and we're going to see wild oats. Year two, after we hayed it, we haven't tilled it, we haven't fertilized it, we haven't done anything. We just let nature do its thing. So now we go from a 0.2 to maybe a 0.35. Those wild oats now have the signal not to grow. As we have that pasture in stand, we don't see wild oats because we keep it above that 0.3. When we decide we want to break it up and we go and we're going to spray it, we're going to disc it, and we're going to fertilize it, that brings that fungal to bacteria ratio down, because as we do tillage, as we add fertilizer, as we add fungicides, that kills off fungi, so that drops our fungal to bacteria ratio. When we do that, and the more intense we do it with our vertical tillage and diskers and high rates of fertilizer, that really drops our fungal to bacteria ratio. And then after 2 or 3 years, we start seeing wild oats. So if there was a way, when we're breaking up a pasture or a hay, if there's a way to support mycorrhizae, fungi, that that would slow down the process of the decline of our fungal to bacteria ratio. So that's one of the challenges. Another way of looking at it is this kind of diagram, where on the left we have our annual weeds and our crops, and then we have our perennial grasses and weeds, then we have our shrubs, then we have our aspens, then we have our mature forest. And as we go to the left, we're bacterial dominated. As we go to the right, we're fungal dominated. The other thing that happens is, when we're bacterial dominated, in our soils, we are nitrate dominated. In a forest, we are ammonium dominated for our nitrogen source. So anytime I see a soil test with high nitrates, I know that the fungal to bacteria ratio is low, and they did not have something green growing in the fall. So it's some really neat tips and of seeing, and once again, high nitrates, there's going to be a high weed pressure. The other ratio that I find really important is knowing your carbon to nitrogen ratio, and knowing exactly where it is. Not a real big deal, but just understanding this carbon to nitrogen ratio and how it works. So this is going to be the ratio of the mass of carbon to nitrogen in residue. So when we have a wide ratio, so anything over a 24 to 1, that's going to be a slow rot. And so in the picture, that's going to be that straw. When we have a narrow carbon to nitrogen ratio, so less than 24 to 1, we're going to have a quick rot. And that's where those green plants tend to have a tight C to N ratio. That 24 to 1 ratio will maintain ground cover. So in that picture, we're dealing with an 80 to 1 on the straw, and those little baby tillage radish, those are going to be probably 6 to 1. Just to kind of give you a visual. So the slow decomposition, it relies on fungi. So using fungicides is going to slow down any residue rot. So it's going to take out a lot of the saprophytic fungi. The positive is of the wide C to N ratio is it maintains soil armor, so it keeps that ground protected. But it is a slow release of nutrients. So once again, you're not going to get a big burst. It's going to be rotted down throughout the whole growing season. The fungi are very slow growing and very sensitive to disturbance. They're sensitive to chemicals also. Now, when we start talking about this fast rot, it's going to be dominated by bacteria. Bacteria can increase their populations tenfold in 4 days if the conditions are right. These are going to cycle those nutrients real quickly, And in this case, this is a tillage radish that I seeded in the fall, and it was about knee high for the top growth. And this picture was taken the end of May, and it is rotted down, and all those nutrients are into the soil. Now, when you look around it, there's no plant growing. So with those free nutrients sitting in the soil, now they're subject to loss, to get tied up by the soil chemistry, or in the nitrogen source, getting converted back to nitrate, and we're going to get a flush of weeds. In this case, it just happened to be between two rows of some winter triticale, so you don't see the triticale, but yeah, it's there. So this is just a quick review of regenerative ag, and each operation, each farm is going to have a little different path, whether you're cash cropping, whether you're mixed farm or livestock. But there's also an opportunity to cooperate. For us on our farm, we don't have livestock, but I want that animal impact. So for the last 20 years, what we're doing is growing grazing corn and bringing the neighbor's cows over. He allowed me to feed his cows for that would be 5 months. And so I got all the manure. He actually paid me cash for custom grazing, and it worked. Now what we were doing is we're seeding full season cover crops, and now we're custom grazing bison. So it helps diversify the farm a little bit more. And right now, like during the winter, where a traditional grain farm, their land isn't making their money, and for me, I'm getting grazing income off of it. Never mind the fact that I'm getting all that manure back on the ground. I'm have that diversity. It's a big win for me. Never mind for the livestock producer. In order to reduce inputs, you must earn the right to do so. And the way we earn the right to reduce inputs is, number one, we need to build micropopulation and their diversity. And how we do that is, number one, is we need to increase the plant diversity. So whether it's, you don't have to go crazy and have all these plants growing and still trying to grain farm, but we can pick species that are going to grow underneath that cash crop, or going to the next level of intercropping, which we'll touch on a little bit. We want to reduce our tillage, and once again, it's to do tillage isn't a bad thing, but what is important is the intensity and the duration. So if we are going out and doing tillage, number one, why are we doing it? Number two, what are we doing to change? Like, what are we trying to change? And what else are we doing? So are we doing some tillage because we're going to be seeding a cover crop? Are we adding more diversity? Are we putting down some humates? What are we trying to repair? Adding this livestock integration, another great way of increasing that microbial population, with between the saliva of the animals, the hoof action, and all the manure and urine. That's dollars. That's dollars going back into the ground to feed that next crop. And by doing that, it's going to help reduce the use of synthetics. When we're using fertilizers, right now, if we did a Sparks test or a Reams test on your soils, Elaine Ingham says that we have between 1,500 and 2,000 years of nutrition in our soil. So when we have a nutrient deficiency, that's because we have a microbial deficiency. So when we use fertilizer, what we're doing is we're taking a job away from the microbes and putting a synthetic in the soil. So the microbes will go in and they'll use it, but we've really reduced the diversity of those microbes in our soil, because there's going to be some other species that, oh, okay, I don't have to work because we're getting free nutrients. The other thing about fertilizer, make sure when we're putting down any fertilizer, make sure we're also adding a carbon source with it, whether it's sugars, whether it's humates, things like that. We need to balance it, because there's some reports of, for every ton of nitrogen fertilizer we use, we're going to be burning off about 30 metric tons of CO2. Because as a carbon-based life form, any nutrition we eat is always associated with carbon. When we are feeding our soil, when we're putting down NPKs, there is no carbon. So that carbon has to go through the microbes, and the microbes, if we don't have the diversity of root exudates, way the microbes are going to get the carbon is through organic matter. So they're going to eat the organic matter to convert the fertilizer, and then the plant can get it. But by that time, we've already taken a hit on the carbon side in the organic matter. So that's the fertilizer. When we start talking about the sprays, the herbicides. Herbicides, once again, read your weeds. What are those weeds telling you? So when Weeds Talk by J.L. McCammon, uh, I should be getting a big royalty check from them, because every presentation I do, I tell people, go get it. To find out what conditions are in the soil that are triggering those weeds to grow. So let's take that ecological advantage away from those weeds. Fungicides. We use the fungicides, we're nuking our fungi. So we need to move away from that. So those are the some of the evils of synthetics. Never mind the insecticides that are, they'll just fry everything out there, and basically the insects are the garbage men of nature. So when that plant is diseased, it's because, and then the insects come in, it's because the plant is right. It's sick, and it's sick because our soil microbiology is so far out of whack. So intercropping. So intercropping is the process of growing two or more cash crops together. So some of the more popular ones are peas and canola, oats and peas. Lots of people using that. Flax and oats. It was something that when I was researching the Saskatchewan Field Husbandry Association was a thing back in the 20s and 30s, 1920s and 30s. My grandfather was a member of, so I did a little bit of research on it, and they were at one of the reports I read was trying to figure out what was the best rate of oats and flax to grow together, and they thought about 2 bushels of oats and 10 pounds of flax. This way, the flax straw is going to be easier to handle. They're both mycorrhizal. Bingo bango, Bob's your uncle, and you're in business. In this picture, this is a picture of an organic farm down in Moose Jaw, and this is flax and chickpeas. And he said the year before, this whole field was just covered in wild mustard. And you can see in the picture, there's one there, but he said there was maybe 12 wild mustard plants in the whole field. So once again, you throw a little bit of Italian ryegrass in there as a grass. So with the chickpeas, there's your legume. Your broadleaf is your flax, and the Italian ryegrass is your grass. Once again, you got the triangle figured out. The key is, number one, is picking seeds of different sizes so they're easy to separate. So if you're going to try growing flax and canary seed, and it's not going to work unless you're going to be feeding it, and you don't have to separate it. But make sure there's good seed size separation, and also talk to your buyers. So in the case of oats and peas, talk to your buyer to say what happens if there's some peas in the oats and maybe a little bit of oats in the peas. Is that a big grading issue? The other thing is we want to pick plants from different functional plant groups. So in this case, once again, your grass, your legume, your broadleaf, pick those. One of the big keys in my mind is making sure you have a legume in there so that there's your nitrogen source. So in the flax and oat, okay, so we're missing a legume there. And once again, because you can't always grow peas year after year, in this case, maybe the subterranean clover is going to be the thing you want to use. So a low-growing clover, be fixing nitrogen, mycorrhizal, so you once again have that triangle all cleaned up. So yeah, the legumes will supply some or all of the nitrogen. So this is going to go back to density. My quick rule of thumb is when we start thinking about percentages and blends, when we start looking at, and if we used subterranean clover, for example, and we said that a pure stand of subterranean clover, which is about 10 pounds, is going to fix about, once again, easy math, 100 pounds an acre. So if we're using 2 pounds of subterranean clover, then, as a quick budget, you should be able to budget on about 20 pounds of nitrogen for that crop. In reality, because of the mycorrhizae, if we get that mycorrhizae cranking and we have that legume that fixes nitrogen, and we have the grass, and the root system will accumulate phosphate because of it can it just covers a bigger volume. You get the mycorrhizae linked together. In order to fix nitrogen, the legume needs lots of phosphate. So the mycorrhizae will accumulate phosphate both from the phosphate solubilizing bacteria it associates with, but also from the grass, moves that into the legume. The legume fixes nitrogen, then moves that nitrogen over to that grass. So we have that system going with those legumes also. So if we're going to be growing peas and canola, there's no mycorrhizal bridge between them. But what'll happen is the peas will keep leaking, uh, these high nitrogen exudates into the soil, and the canola roots will grow into it, and it'll take that away from that pea plant. So when the pea senses the nitrogen around their roots is dropping, it just keeps out leaking more and more of that nitrogen. So this is how it feeds that canola plant. And the other thing is that canola plant has a very acidic root system, so then it's going to be able to break some of these calcium phosphate bonds, so you can actually get away from getting from using lots of fertilizer with a pea canola mixture. So when you look at some of these other broadleaves, like say sunflowers, they have some other ecological advantages, like whether it's saline tolerant, maybe a little more drought tolerant. So play around with some of these species, and knowing their seed size is going to be one of the keys. So the regenerative ag culture, each operation is going to be different. Whether it's their goals, what assets they have, what issues. There's no cookie cutter answers when we get into this. We can guide, and we can generalize, but how you're going to do it is going to be different from farm to farm. But to use as many of the 5 regenerative agriculture principles as possible, that's the key. And the number one, if you only decide you want to do one of them, if you keep that living root of the vegetative plant throughout the whole growing season, that's one of the easiest, because using the subterranean clover, which is low growing clover, you want to go into a perennial, into a cropping system, use some white Dutch clover as 2 legumes. You get into your grasses, you can use winter cereals. There's a whole bunch of different ways of getting it done. And the other thing that I highly recommend is, as young people starting your careers, challenge how things are being done. The scariest phrase in any business is, we do it this way because it's always been done that way. Challenge it. Ask questions. So if you're interested in doing some more reading, my recommended reading list, Mycorrhizal Planet by Michael Phelps, really good book. The Farm as an Ecosystem by Jerry Benetti. Quality Agriculture by John Kempf. The Teaming With series, there's 3 of them: Teaming with Microbes, Teaming with Bacteria, Teaming with Fungi. Really good read. Call of the Reed Warbler, just finished reading that one. That's a good one. The 3 books by David Montgomery, excellent reads. Fundamentals of Soil Ecology, really good read. And then When Weeds Talk by J.L. McCammon. So hope you enjoyed it, and if you have any, if you want to reach out, there's my contact information, and have a good one.